Biology 210, Lab 9, Exercise 25, the standard plate count. In Exercise 25, serum dilutions and the standard plate count will be performed to calculate the original cell concentration, or OCC, of a sample of E. coli. Before you begin Exercises 25 and 26, view Lab 9, Exercise 24, Introduction to Dilutions YouTube video, and complete the practice homework problems on pages 225 through 227 of the lab manual. You should definitely view these before you view this particular video, because otherwise this video will not make sense. So in the first video, we talked about how to set up dilutions and why we do them. In this video, we're going to actually apply what we learned in the dilution introduction. Exercise 25, Standard Plate Count Method, page 229. So here you see a test tube of E. coli. Let's talk about what we're going to do with this test tube of E. coli. If a bacterium, such as E. coli, is provided the correct growth temperatures or conditions, temperature, pH, salinity, etc., then it is possible in a short period of time the population of the bacterium will grow quickly and become very dense. For example, if E. coli is in the proper environment, then its generation time, which is the time it takes for the population size to double, is 20 minutes. Once inoculated, a broth of E. coli contained millions or billions or trillions of cells in 24 to 48 hours after incubation. So what are we going to do within this experiment? The purpose of this exercise is to dilute a sample of E. coli, perform the standard plate count method, and calculate the OCC. The purpose is to determine the number of E. coli cells in the test tube. So how do you know by looking at this slide of E. coli that it is actually going to have lots of cells in it? Correct, it is cloudy or turbid. So we're going to dilute this sample, which is heavily, densely populated with E. coli, several times to get to a number between 30 and 300 colonies. So how many cells of E. coli do you think is in this test tube? Write your hypothesis on page 231. At the end of this, I will give you hypothetical data and we'll see if you're close. It's kind of like taking a jar of jelly beans and guesstimating how many you think there's in the jar. Exercise 25, Standard Plate Count, page 230. So on page 230, it is going to talk about what is the standard plate count. It is a method used to dilute a dense population of bacteria. It is used to determine the number of bacteria in a sample. After a sample is diluted, varying amounts of the dilution are added to a plate or plates, agar is added, and colonies form. Recall that it is important to have agar at a certain temperature. If the agar is too hot, you will kill all the bacteria you're trying to grow. If the agar is too cool, it will solidify. After incubation, the number of colonies are counted. The original cell concentration, or OCC, is then calculated. Again, what that means is we can figure out how many almost exact E. coli cells are in this test tube. Each colony on the plate represents viable cells. Viable cells are living cells. Only plates between 30 and 300 colonies, which we call the magic number, are counted or calculated. Keep that in mind as we proceed through the experimental data. Exercise 25, Setup of the Experiment. Figure 25.3 on pages 230-231. So we're going to go through step by step of how we set this up and why we do this. Step 1. A sample of E. coli is pipetted into a water dilution bottle containing 99 ml of water. So we call those bottles dilution bottles or water bottles. They contain 99 ml of water and they're used for dilution. Remember, this is very similar to what we did several weeks ago with agar deeps. We just have exact amounts of 1 ml and 99 ml for this experiment. A serial dilution is made. A serial dilution is a series of dilutions or bottles in which we get more and more dilute. So for example, bottle A will have the most bacteria, bottle B will have less, and bottle C will have the least. But we don't care about how much is in the bottle. We want to plate those amounts and then grow the bacteria to C colonies. So then we take 1 ml or 0.1 ml and we pipette them into plates. We add nutrient agar, and we allow them to solidify. We allow them to grow and incubate them at their proper temperature of 38 degrees, and after 48 hours, we see colonies forming on the plates. We will not be able to tell on certain experiments what bacteria are present, just that bacteria are present. In this experiment, we know it is E. coli because we started with the test tube of E. coli. Note in red, it says, new pipettes are used for each transfer between dilution bottles. So every time you go from bottle to bottle, a new pipette must be used. When you go from bottle to plates, a new pipette must be used. We don't want to cross-contaminate any of our samples. Exercise 25, setup of dilution bottles, page 230. So on 230, we're going to break this into small bits. Dilution bottle A is going to have a dilution of 10 to the negative 2. Let's talk about why that is. At the bottom of the screen, we see the formulas. It is very important for a test or quiz that you know the formulas and examples and how to calculate these for quizzes. So let's look at our first formula, single bottle. The volume added, in this case, for every single bottle, it is 1 milliliter. We're going to use pipettes for that. Divided by the total. In this case, for all of them, it's 99 mils of water, and we're adding 1 mil. So 1 plus 99 is 100. We use 1 over 100 because it's an easy and fast number. Recall that 1 over 100 is a fraction, which is the same as 10 to the negative 2. It's 10 to the negative 2 because it is a negative exponent, or a fraction. It's 2 because there are 2 zeros in 100. So every time we take 1 mil and put into the next bottle, it is a 10 to the negative 2, or 1 over 100 dilution. Those are then accumulative or summative. So our series of dilution bottle formula is dilution of in bottle times dilution of in bottle. Remember that when we multiply numbers with exponents, we add the exponents. So if we have 3 bottles that are each 10 to the negative 2, that would be 10 to the negative 2 times 10 to the negative 2 times 10 to the negative 2. When we add exponents, 2 plus 2 plus 2 gives us 6. So that very first bottle has a dilution of 10 to the negative 2. Our second bottle, bottle B, has a dilution of 10 to the negative 4. Our third bottle, bottle C, has a dilution of 10 to the negative 6. So each bottle is more dilute than the previous bottle. Again, we want to shake up the bottles and then remove pipetting them into plates. Exercise 25, setup of plated dilutions, page 230. So now we're going to talk about the component of adding various amounts from the bottles into plates. So 1 mil and 0.1 mil are pipetted from dilution bottle B into plates. The plated dilutions from dilution bottle B are 10 to the negative 4 and 10 to the negative 5. Let's talk about why that is. Again, refer to the formula. Our plated dilution is the volume plated. Our choices for that are 1 mil or 0.1 mil times the dilution bottle. So in this case, dilution bottle B has a dilution of 10 to the negative 4. We can withdraw a pipette 1 mil. 1 times 10 to the negative 4 is 10 to the negative 4. Our other plate gives a 10 to the negative 5 dilution. How did we get that? 0.1 mil times 10 to the negative 5, 0.1 mil times 10 to the negative 4 gives us 10 to the negative 5. So how did we get that? So 0.1 mil is the same as 10 to the negative 1. So 10 to the negative 4 times 10 to the negative 1 is 10 to the negative 5, which you can see at the bottom of the screen. Our third bottle, bottle C, also has 1 mil and 0.1 mil pipetted into plates. So let's see how we got those plated dilutions. 1 was pipetted out of the 10 to the negative 6 bottle. 1 times 10 to the negative 6 is 10 to the negative 6. 0.1 was pipetted out of the 10 to the negative 6 dilution bottle. 0.1 times 10 to the negative 6 gives a 10 to the negative 7. So which bottle or plate is going to be the least dilute? Correct, 10 to the negative 7. So what we're going to see in a moment is the actual amount of colonies that grew on each plate. So let's look at each plate and see what you think is going to happen. We have 10 to the negative 4, 10 to the negative 5, 10 to the negative 6, 10 to the negative 7. So we get more dilute as we progress, which means less colonies should form. Also think about, why did we not use the 10 to the negative 2 bottle to actually form plates in this experiment? We will answer that later when you get to the conclusion portion in your lab manual. Exercise 25, data results, page 232. On this slide, you will see the results from our experiment. Our 10 to the negative 4 plate has the most colonies as predicted. It has over 300 colonies, or too numerous or too many to count. Our 10 to the negative 5 plate has 295 colonies, which falls within our range of 30 to 300 colonies. Our 10 to the negative 6 plate has 97 colonies, also countable. And lastly, our most dilute plate has the least amount of colonies at 26 colonies, or too few to count. Using the data below, write the amount of colonies on the plates on page 232. So all you're going to do is on page 232 of your lab manual, transfer the numbers that you see on these plates onto the table in the lab manual and on the plates. Using the data below, fill in the table on page 232. So you're just transferring the data from this slide directly onto the lab manual pages. You're going to then use these numbers to calculate the OCC, which we'll go over the next few slides. Calculating the results, page 232. So let's recall our formulas and go through some examples. So we are not going to use our 10 to the negative 4 plate because it was too many colonies to count, over 300. We are not going to use our 10 to the negative 7 plate because it was too few, only 26 colonies. So we're going to focus our attentions on the 10 to the negative 5 and 10 to the negative 6 plates, which contain 295 and 97 colonies. So let's review what formula we need to use for this experiment. We want to calculate the original cell concentration. That is how much was in that original test of E. coli. Our equation is the number of colonies, which is either 295 or 97, times the dilution factor. Recall, the dilution factor is the reciprocal or the inverse of the dilution. The dilution, 10 to the negative 5 in this case, or 10 to the negative 6, is always going to be a fraction. And because the dilution factor is the reciprocal inverse, it is always going to be a positive exponent, or 10 to the 5 or 10 to the 6. Dilutions, negative exponents. Dilution factors, positive exponents. So up above in the example, I go through how to calculate this. So for example, if we had 52 colonies on a plate and our dilution was 10 to the negative 5, our dilution factor would be 10 to the 5th. So we'd take 5.2 times 10 to the 6th. That would be our final answer. How did we get that? 52 colonies that we counted on our plate times 10 to the 5th, which is our dilution factor. We need to move the decimal one place to put it in scientific notation. Moving the decimal one place gives us 5.2 times 10 to the 6th. That would be the final answer. Exercise 25, conclusions and analysis, page 233. This particular slide goes through everything that we've done so far today, how we set up the experiment and the results and the colonies that formed on the plate. So what we can do is calculate how many E. coli cells was in that original test tube. Did you guess the number of E. coli cells in the sample correctly? Was your hypothesis correct? I hope so.